Alright, today we are going through the process of using a hemocytometer for using cell counts if we need to do an experiment that requires a certain number of cells. We'll use this process to get cells here in the core biology facilities. So the first thing I'm going to do is, if you come over here, we have a small flask of cells that we trypsinized and then resuspended in media. So that's essentially just took all the cells out of a flask and put them in this vial. What you're going to do is you're going to grab the 20 to 200 microliter micropipette, and you're just going to take out 100 microliters. So I'm going to dial in to 100. Alright, so we're going to take an Eppendorf tube like this, and then we're going to take our cells. We're just going to take 100 microliters. Squirt it into there. Alright. Next, get rid of our Eppendorf tube and actually move into the area we're going to work. So we're using Trypan Blue as our stain, and all we're going to do is take 100 microliters of this as well and put that in the Eppendorf tube. So alright, so now we have our cells ready. You can even resuspend and stir them up a little bit, but mostly they're good to go. Okay, set that off to the side. Make sure it doesn't leak. Alright, so now we have our hemocytometer grid, and we're going to want to make sure it's clean, so we'll give it a little spray of ethanol. Just a little tiny amount will do. Make sure you get the working surface. Your working surface that you're dealing with is between these red marks, and then we have a little cover slip of glass that will also give a little bit of ethanol. So you can wipe these down. Just take a Kim wipe or two, dry them all off, make sure everything is clean before we get started. Okay, so you're going to take your cover slip once it's dry. It should dry pretty quickly. And you're going to set it right on the working surface here. Right on it. Okay. Your next step is going to be to grab another micropipette tip. And you just want a tiny amount, so you don't even need that full 100 microliters. So just take kind of an arbitrary amount. But what you're going to do is you're just going to take a small amount into your tube. I just took just a tiny amount. And then you're going to insert the tip of your micropipette right alongside the cover slip here. There's a little groove between the red lines that are parallel to each other. And you're just gonna squirt the smallest amount until it hits. You'll see the fluid come over and into the working surface right here. So once you have enough where that has happened, you can go and bring your slide up and into the microscope and turn on your microscope. So the first thing you're going to see when you look under the microscope is a grid somewhat like this. you're only going to be able to see a field of view something around this square. So as you can see, there's 4 sections that have the larger squares. And what you're going to do is you're going to count how many cells are in these 16 blocks, and then you're going to move over, count how many cells are in these 16 blocks, and do that for all 4 corners. And there's a tool right here that you can use to count each block individually. And you just have to count up on each one as you go. The 5th block you can use for any cells that are stained really dark. Those are your dead cells. So you want to use this counter, the 5th one, to count how many dead cells you have total. So at the end, when you do viability tests, you understand how many cells are not living. All right. So now that we have taken our counts and we have 4 separate numbers, what we want to do is you want to take an average of those numbers, so add them together and divide by 4, and then you will multiply that number by 2 to account for the dilution of the trypan blue. And after you have that number, it will give you that number times 10 to the 4th cells per milliliter. So if the number is 72, it's 72 point whatever times 10 to the 4th cells per milliliter. And you can use that number to know how many cells you want to put in each experiment that you're working with. So when you're done making all your calculations, using your stuff, just make sure your workstation's clean. All you need to do is pull this off, pull your little cover slip off. You're going to want to wipe it down, make sure all the cells are off of there. Hopefully the next person will use ethanol to clean it up even better. And there's a little container in the drawer down here that you can put your cover slips into. So we can drop your cover slip in there, and then just make sure your plane goes right in the box.